Here we have the phospholipid. The phospholipid structure contains two different sections. The first section is the phosphate head, which contains a phosphate group. This group is connected to a glycerol which bonds the head to the tail. The tail consists of two fatty acid chains. This one is saturated and this one is unsaturated due to a double bond in the chain. Due to their structure, the head is hydrophilic and the tail is hydrophobic. Phospholipids can be oriented in three different ways when exposed to an aqueous environment. The hydrophilic head will always be exposed to the solution while shielding the hydrophobic tail from exposure. Here we see a lipid bilayer, also known as the plasma membrane of a cell. Its role is to control what is able to exit and enter the cell. Embedded proteins are utilized in order to transport polar proteins and molecules that have difficulty penetrating the bilayer. The protein is able to embed themselves into the bilayer because its ends have hydrophilic R groups in their amino acids, while the insides have hydrophobic R groups. Surface proteins attach to the outside of the membrane. They're able to do this because the R groups in their amino acids are hydrophilic, giving the protein the ability to be exposed to water and creating attraction to the outside of the membrane. The locations of the cell's surface proteins function as markers on the cell to help cell-to-cell recognition. The cell membrane is described as a fluid mosaic because of the many components that contribute to the fluidity of the membrane. These components include glycoproteins and glycolipids, carbohydrates covalently bonded to proteins and lipids, and their role is to aid in cell-to-cell recognition. Next are embedded proteins, which as mentioned before are necessary to allow passage of hydrophilic substances through the membrane. And finally, the role of steroids, such as cholesterol, is to reduce phospholipid movement, which in turn reduces fluidity. The plasma membrane is able to do its job due to selective permeability. This means the plasma membrane and its component molecules work together to allow certain substances to cross in and out of the cell. Polar molecules or ions that are hydrophilic aren't able to pass freely due to the hydrophobic interior of the membrane. A charged molecule is even less likely to be able to penetrate the bilayer. Large polar molecules are not able to pass. Only the ones that have functional carbon groups may pass, but with great difficulty. Even though they're polar, these molecules are able to pass with less difficulty than the previous molecules due to their small size. An example of one of these molecules is water. These molecules are able to pass through freely because they're small and not polar, so they can mix well with the nonpolar phospholipid layers. A cell wall is another way the cell can regulate what substances come in and out of the cell. The cell wall also gives the cell strength and structure. Diffusion is the movement of molecules from a highly concentrated region to a less concentrated region. Osmosis is the diffusion of free water across a selectively permeable membrane. Facilitated diffusion is the passage of molecules or ions down the electrochemical gradient across a biological membrane with the assistance of a specific transmembrane transport protein. These three modes of transport are examples of passive transport, which means that there is no energy required. The movement of molecules from a low concentrated region to a highly concentrated region is defined as active transport. This requires embedded proteins and energy from ATP. A special case of active transport is the sodium-potassium pump. With energy from ATP, the pump moves sodium and potassium in opposite directions against the concentration gradient. In one pump, 3 sodium ions exit and 2 potassium ions enter the cell. Exocytosis is the transport of large molecules out of the cell, and endocytosis is the transport of large molecules into the cell. Both require ATP.